Introduction. Today, we will explore the fascinating world of histidine, a vital amino acid with crucial roles in various biological processes. Histidine plays a significant role in protein biosynthesis, immune function, histamine synthesis and metabolism, and coordination of metal ions, among others. Furthermore, we will discuss histidine-related disorders, histidine transport systems, catabolism, and its potential as a precursor for biologically active molecules. So, let us dive into the intricate pathways and functions of histidine within the human body. Histidine is an essential amino acid that cannot be produced in the human body, making it necessary to obtain from dietary sources. The biosynthesis of histidine begins with the precursor molecule 5-phosphoribosyl 1-pyrophosphate, PRPP. This crucial step is catalyzed by the enzyme phosphoribosyl pyrophosphate synthetase 1, PRS1. Subsequently, PRPP undergoes a series of reactions involving several enzymes, including phosphoribosyl AMP cyclohydrolase, HISI, histidinyl phosphate aminotransferase, HISC, and histidinyl phosphate phosphatase, HISA. These enzymatic reactions enable the formation of histidinol, which is further converted into histidine by histidinyl phosphate transaminase, HISP, and histidinyl dehydrogenase, HIST. The biosynthesis of histidine is tightly regulated, ensuring the production of appropriate levels to meet physiological demands. The biosynthesis of histidine is regulated through a process called feedback inhibition. In this mechanism, the end product of the biosynthetic pathway, histidine, binds to the enzyme histidine biosynthesis sensor protein, HBSP. This binding triggers a conformational change in HBSP, leading to the repression of gene expression for histidine biosynthetic enzymes. Moreover, the regulation of histidine biosynthesis involves multiple transcription factors, such as HISR and HIS, which modulate the expression of genes involved in the biosynthetic pathway. Environmental factors, including the availability of other nutrients and the presence of metabolic intermediates, also influence the regulation of histidine biosynthesis. Understanding the intricacies of histidine biosynthesis regulation is crucial for developing therapeutic strategies for histidine-related disorders. Histidine-rich proteins, HRPs, have shown to play vital roles in immune function. These proteins contain a high concentration of histidine residues and are involved in various immune-related processes, including pathogen recognition, antimicrobial activity, and modulation of immune cell signaling. HRPs, such as histatins and S100 proteins, exhibit potent antimicrobial properties, contributing to innate immune defense against invading pathogens. Moreover, histidine-rich domains present in proteins like toll-like receptors, TLRs, participate in pathogen recognition, initiating immune responses. The intricate interplay between HRPs and immune function highlights the importance of histidine in maintaining a robust immune system. Histamine, derived from histidine, is a biogenic amine involved in various physiological processes, including allergic responses, gastric acid secretion, and neurotransmission. Histidine decarboxylase, HDC, catalyzes the conversion of histidine into histamine. Histamine metabolism primarily occurs through two main pathways: the oxidative deamination pathway, catalyzed by diamine oxidase, DAO, and the methylation pathway, involving histamine N-methyltransferase, HNMT. These enzymatic reactions ensure the maintenance of histamine homeostasis within the body. Dysregulation of histamine synthesis and metabolism can contribute to various histidine-related disorders, emphasizing the importance of maintaining proper histamine levels for overall health. Several disorders can arise due to disturbances in histidine metabolism. One such disorder is histidinemia, resulting from a deficiency in the enzyme histidase, responsible for histidine catabolism. Histidinemia is characterized by high levels of histidine in the blood, which may lead to developmental delays, intellectual disabilities, and behavioral issues. Furthermore, histamine intolerance, characterized by an impaired ability to break down histamine, can result in symptoms such as migraines, gastrointestinal disturbances, and allergic-like reactions. Better understanding of these histidine-related disorders contributes to the development of diagnostic approaches and potential therapeutic interventions. The transport of histidine across cellular membranes is essential for maintaining proper histidine levels within cells. Histidine transport systems encompass multiple transporters, including the solute carrier family 22-member 4, SLC22A4, and solute carrier family 22-member 5, SLC22A5. These transporters utilize various mechanisms, such as the facilitative transport of histidine, to ensure its uptake by cells. Dysregulation of histidine transporters can disrupt histidine homeostasis, impacting overall cellular function. In-depth knowledge of histidine transport systems contributes to understanding cellular uptake mechanisms and potential avenues for therapeutic interventions. Histidine catabolism involves the breakdown of histidine into various metabolites. The initial step of histidine catabolism is catalyzed by histidine ammonia lyase, HAL, which converts histidine to urocanic acid. Subsequently, urocanic acid undergoes further enzymatic reactions to form imidazole propionic acid, IPA. IPA can be further metabolized into several compounds, including beta-alanine, which serves as a precursor for the synthesis of pantothenic acid, a vital component of coenzyme A. Understanding the catabolic pathways of histidine aids in discovering the potential roles of its metabolites and their significance in various physiological processes. Histidine serves as a precursor for the synthesis of biologically active molecules, including carnosine and histamine. Carnosine, composed of histidine and beta-alanine, possesses antioxidant and anti-aging properties. It also functions as a pH buffer, contributing to muscle function and performance. Histamine, derived from histidine via HDC, acts as a neurotransmitter involved in synaptic transmission, sleep-wake regulation, and immune responses. Exploring the diverse roles of histidine-derived molecules sheds light on the multifaceted functions of this amino acid within the body. Histidine plays a vital role in metal-ion coordination, acting as a ligand in metalloproteins. The imidazole side chain of histidine can coordinate with metals such as zinc, copper, and iron, enabling various biological functions. Histidine residues participate in the coordination of metal ions in enzymes, facilitating catalytic reactions. They also contribute to metal transport, storage, and sensing processes within cells. Understanding the role of histidine in metal ion coordination enhances our comprehension of essential biological processes involving metalloproteins. Emerging evidence suggests that histidine metabolism is dysregulated in various cancers. Altered expression of enzymes involved in histidine biosynthesis, such as PRS1 and His, has been observed in cancer cells. Moreover, histidine catabolism and histidine-derived metabolites, such as carnosine, have been implicated in cancer pathogenesis and progression. Investigating the intricate mechanisms underlying the regulation of histidine metabolism in cancer provides valuable insights for novel therapeutic strategies targeting this vital amino acid. Histidine supplementation has gained attention for its potential health benefits. Studies have suggested the positive effects of histidine supplementation in promoting wound healing, preserving muscle mass, and improving cognitive function. However, the optimal dosage and long-term effects of histidine supplementation in different physiological and pathological contexts require further investigation. Analyzing the outcomes of histidine supplementation contributes to our understanding of the potential therapeutic applications of this amino acid. Growing evidence suggests that histidine is involved in neurodegenerative diseases, such as Alzheimer's, Parkinson's, and Huntington's disease. Histidine-derived molecules, including histamine and carnosine, exhibit neuroprotective properties by modulating oxidative stress, inflammation, and protein aggregation. Additionally, dysregulation of histidine metabolism and histidine-rich proteins may contribute to neurodegenerative disease pathogenesis. Exploring the role of histidine in neurodegenerative diseases provides insights into potential therapeutic strategies for these debilitating conditions. In conclusion, histidine plays a fundamental role in various biological processes, ranging from protein biosynthesis to neurodegenerative diseases. Understanding the biosynthesis, regulation, and metabolism of histidine, as well as its diverse functions, is crucial in unraveling its significance in health and disease. Moreover, the exploration of histidine-related disorders, transport systems, and histidine-derived molecules broadens our understanding of histidine's intricate involvement in maintaining homeostasis and supporting vital physiological functions. Continued research in the field of histidine biochemistry offers promising opportunities for developing targeted therapies and interventions for various disorders. As we uncover more about the complex mechanisms and functions of histidine, we take steps towards harnessing its full potential for human health and well-being.